Hello, everyone. Welcome to our webinar. Good afternoon to Europe and good morning to America. We have something, and common. This one will be a little bit shared with the EPA, and therefore I will give Holly a couple of minutes later to introduce herself. I'm Thomas Exner. I'm the coordinator of OpenRiskNet. I will first give a short introduction to OpenRiskNet, and then we will hopefully hand over to Marvin to present the one case study. On the screen, you are seeing at the moment the series of webinars we had already and we will have in the next future with a couple of already recorded webinars on the case studies, but also on technical details on OpenRiskNet, and we will continue in that way. Today we will talk about integration of data and the relation to AOP-Wiki, and then in 2 weeks' time we have a follow-up which then describes the adverse outcome pathway database. As I announced, this is combined with the EPA, and therefore, Holly, if you want to give a short introduction to yourself and to your group, you are welcome to do so.
All right, so thank you for letting me introduce myself. I'm Holly Mortensen. I work at the National Health and Environmental Effects Research Lab of the EPA. I'm a research scientist here. I'm in the room currently with my 2 students, Philip Langley and Trevor Levy, as well as Brian Chorley, who is involved with the Adverse Outcome Pathway Discovery and Development task here at the EPA, and Mark Nelms has joined us from NCCT, another part of the EPA as well.
Okay, thank you very much. Later, Marvin will probably describe also this interaction between these two groups in his presentation. But as I said, first I want to give you a short introduction of what OpenRiskNet tries to do. This is an extract from our proposal. That when we started with the project, we were just all a little bit frustrated with all these different tools which are available, but which are not really in the standard way to access all these different databases, which you more or less have to query by hand. And to make that easier, to also deploy these tools easier, OpenRiskNet was trying to develop this set of tools which interacts, or which first gives you an infrastructure where you can run your calculations, but then also get the tools easy in there. What we are doing, and this is what we present here mainly, is this case study driven approach that we try to see how risk assessment can, or the different parts of risk assessment, how these tools we develop can help these tasks in risk assessment, and how you can combine that from data collection to then the final result. We are concentrating on, or we are relatively broad, chemical and nanomaterial risk assessment, and we try to build these cases around that. And then main point is really here that we want to integrate the existing tools, not develop new tools, but develop the infrastructure where then these existing tools can be put on, and in that way be used easier. These are the case studies we had already, and one of the webinars were on this data curation case study, data cure, which really is focusing mainly on the how to get data from different sources, and in that way bring that into the system. And now we are going to the other case studies where then this data is used, and for example here in the AOPLink or case study, where this is then really used to link to also the AOP-Wiki, and the information is taken there. We have then some bioinformatics, toxicogenomics case studies. We have a QSAR read-across case study, and then PBPK, kinetics modeling, and metabolism. This is just the overview what we have. And as I said, these webinars are also meant to describe these one after another. Therefore, I won't go into much detail now because you will see the first or one example in the remaining of this webinar. I just want to give you a little bit an update where we are. We decided at the beginning that we want to use REST services to integrate these, try to harmonize APIs and annotate these APIs with a semantic layer which describes what these services are doing, what input output they produce or they need and produce, and then take all these things, put them into a container and provide them in what we are calling virtual infrastructure, or sometimes also virtual research infrastructures, where all these containers can be deployed. This looks a little bit like that, yeah, that you have your software, you put that into services, then you deploy that on the infrastructure, and then you can access that via different tools. What I have to say right now is that we are mainly here in the script area, that there are many things which we are still developing, which is easier if you really do that by scripts, but we want to move on to more web-based approaches, and then hopefully also get more and more people in there as associated partners, and the EPA is one of these, which then bring also new tools in and make them the whole infrastructure more usable. This is a list of the services, or some of the services we have already. If you go to our reference infrastructure, reference environment, then you can try them out. This is, as I said, where we are at the moment. Many things are script-based, where we use Jupyter to create these kind of workflows where different tools are put together. One slide on the annotation, which is relatively complex, and we will have another webinar on that, how these services will be annotated, but that is more for the developers. For us, it's then important that we can use SPARQL queries to get this information out, what this service really is. Yeah, and this will then also be integrated in the different tools to that these services can communicate with each other. The final goal is then something more like the Squonk notebook or workflow tool, where this all is hidden in the software, and the user can much easier use all these things by just selecting different services and then drag them and combine them by just putting these arrows somewhere. As I said, we are not quite there, but the concepts, I think, are relatively nicely defined, and we can now really build on top of that these services and then also these more user-friendly tools. If you want to try it out, go to our website. You see here the reference environment, which is our deployment of this whole thing, where you can try it out, or you can also deploy that in-house as your own virtual research environment, where you then can also decide on how much resources you need for the calculations. Our reference environment is a little bit limited in that respect, because it's really only meant as a as a test environment where you can see if that is something for you. We also would like to always get your feedback, and we have a couple of support material, especially also this webinar series which we have. That was more or less it. These are the partners. I don't want to spend too much time because I think you are here because of the case study. But I hope it gave you a little bit the idea where we are, that we are not somebody, or that the project and also the case studies are not so much scientifically driven, that we have specific problems we want to solve. We more think about how can these problems be solved in a more general way by providing the tools, and then the user can combine them to really optimize that for their specific problem. That's all from my side. I think I would propose that we do questions later. If you have something, please put your questions into the question box, which you will see in the GoToWebinar application. You can also raise your hand, and then we will try to or we will unmute you, and then you can also give some comments, at least at the end of the presentation, definitely at the end. And if Marvin agrees, perhaps also in between. Good, that was my part, and I would hand over to Marvin.
Okay, so welcome everyone. I am Marvin Martens from the Department of Bioinformatics in Maastricht University. And together with Egon Willighagen and Grace Evelo, I've been working on one of the case studies of OpenRiskNet called AOPLink. Sorry, my voice is a bit cracked. So as Thomas already described, it's one of the 7 main case studies in the project. And basically, we aim for linking information towards AOPs. So the general aim, the general vision in regulatory toxicology and science in general related to risk assessment is that there is a need for a connection between the scientific area, being the scientists that do toxicological studies, and the data that gets generated from there, besides the knowledge, needs to be useful and made useful for risk assessment, making decisions about guidelines about useful about using certain compounds or chemicals. And that's where AOPLink is playing a role as well. In this webinar, I will describe 3 main topics. First of which is the research questions that we can really answer with this case study. Afterwards, I will describe the tools that are deployed in OpenRiskNet that are used to answer these research questions. And on the third, I will talk about the things that we are planning to do in the remainder of this year before the project of OpenRiskNet finishes. So the first part, of course, this case study did not come out of nothing. We looked into the topic of adverse outcome pathways, AOPs for short, and looked into ways to how to extend them and make them more useful on the side of the risk assessors. From which we came to 2 major research questions that are the founding questions of this case study. So question 1, that is what we want to answer, is for a certain AOP, certain adverse outcome pathway, which experimental data is publicly available or is available to support the AOP. And then the other way around, we want to know if we can support an existing AOP from a certain data set that we have. So can this experimental data support an existing AOP? So it's really this connection both ways between experimental data on the one side and adverse outcome pathways on the other side. So in order to understand the whole purpose of the AOPLink, it is essential to know what is really the concept of adverse outcome pathways. Of course, there's many people that already know about it, but some that might need some short introduction, so I included it in this presentation. So the adverse outcome pathway concept, it is a framework that's originally described in 2010 that captures mechanistic knowledge from a toxicological process to support decision making in risk assessment. On the picture on the right, there's a graphical view of how we draw an adverse outcome pathway. We imagine it as a row of dominoes, in which each domino is a key event, a biological event, which is measurable and essential for progression, which starts at the molecular level, causes a chain reaction to go on cellular effects, organ level effects towards an adverse outcome. The first stone that is tipped over by something called a stressor. In many cases, this is a chemical, but can be many more kind of things such as radiation. And it is called the molecular initiating event. That is the first stone. Of course, key events are also connected, and these connections are called key event relationships. There are relationships between each stone, and these are often supported by data, and calculation can be made on response-response relationship. So the concept of the adverse outcome pathway, it is used in a broader concept of the IATAs, the integrated approaches to testing assessment. Basically, they provide knowledge, they provide connections, they provide systems to make testing strategies. Because each key event is said to be measurable, they have information about methods and measurement techniques to test their validity, to test if they really occur. So they have some kind of predictive value to what happens if a chemical or to a system that is exposed to a certain chemical. Therefore, the reuse of information and the gathering of all the knowledge makes the risk assessment more efficient and makes use of animals less prominent, because knowledge already exists and it gets reused instead of recreated. And also, the concept is based on knowledge that is there, so it also is able to identify where knowledge is lacking, where we miss some pieces of information. The main topic of this webinar, AOPLink, the title of it is Linking Data to Adverse Outcome Pathways. So in this case study, we'll look into the linkage of data to AOPs. So as the picture that Thomas already showed of the workflow for safety assessment, this case study in particular covers three main steps in this workflow, which is the tier 0 step 3, collecting supporting data step, the tier 1 step 6, the mode of action hypothesis generation, and the tier 2 step 9, basically the identification of gaps of knowledge. And also a bit similar to what Thomas already described, it's central in this network of case studies in the OpenRiskNet project, where it gets input from case studies such as TGX and SIS group, and the output of the case study, the results that come out of it, are useful input for the model RX case study. So there's an interaction between case studies here, which can be very direct, but also more on the indirect way through data cure and using the data. Now about the tools. We have, of course, already the questions that we wanted to answer, but what tools do we really need to answer these questions? So I have made a global overview that describes the most simple of ways how we envision this case study, which is on one side the AOP-Wiki and on the other side the data. In this case, the databases are WikiPathways, ToxCast, DGGates, and ToxRefDB. I will tell you why later. On the left side is the AOP-Wiki. The AOP-Wiki is the main repository for adverse outcome pathways. I will talk a little bit more about that in a few seconds. So these two sites are the ones that we want to combine, because this allows us to answer the two research questions that we have. As a connection between these two sites, I have put BridgeDb. We have BridgeDb. It's a service that maps identifiers. It is basically the bridge of databases, and it connects the one identifier from the AOP-Wiki to other types of identifiers that are requested in these databases on the other side. And it allows chemical databases to emerge. BridgeDb allows mapping to all kinds of databases. So more knowledge can be gathered from there. Next slide, please. And as I described, it's not only this case study that stands on itself. There's also plans to work with the other case studies, in which the input from TGX and sysgroup can play major roles in here and directly interfere with the workflows that we will create, as well as other projects which I mistakenly mentioned before, the EA-ToxRisk project, in which data is generated and will probably later be used for workflows like this as well. Next slide, please. And finally, we want to extend this workflow and use the data that we get from AOPs or related to AOPs to do pathway analysis. And for pathway analysis, we use PathVisioRPC. It is a pathway analysis tool, which I will also describe later on. So the AOP-Wiki is the most central tool in this case study. The AOP-Wiki is the main qualitative AOP repository of the AOP knowledge base and is a joint effort between the European Commission JRC and the US EPA. It is currently holding 246 adverse outcome pathways with many more key events and key events relationships, as well as 438 stressors, most of which contain chemicals. They are part of this larger AOP knowledge base in which also other parties can play roles, such as Effectopedia, which is more a modeling tool, and AOP Explorer, a cytoscape application. So now I want to describe how the AOPs are currently structured, how they are currently available. So this is one example of an adverse outcome pathway, one of the most famous in the AOP-Wiki, which is the one created by Brigitte Landesman. And this is the general figure that you see in most papers, and it's the first thing that you see when you open the page on the AOP-Wiki website. But there's a lot more information than just that is just described in this scheme. Next slide, please. For example, these pages of adverse outcome pathways, they contain information like the background, the applicability, the key events relationship identifiers that are used for this, the stressors, and the assessment. Next slide, please. The stressors on the besides that, they contain a next level of information, being the types of identifiers, the description, the evidence, but also information about how they link back to the molecular initiating events. Next slide, please. Then you have the key events. The key events are the main keystones, like I phrased earlier, and they also contain a lot of information, which is all on separate pages in this database, and which can be modeled, as adverse outcome pathways are considered to be modular. These are considered as separate entities on the database. Next slide, please. And on top of that, you also have the connection in between them, which I mentioned before as well, the key event relationships, containing many types of descriptive text to describe the support that these connections have, as well as the applicability. So all this data needs to be integrated somehow. And if we want to make this useful, we chose to convert this whole database into an RDF format, for which we made a scheme. So the scheme on the right is the simple version of the adverse outcome pathway wiki RDF scheme, in which all the contents of the AOP-Wiki database in the end is converted into triples, in which all connections make sense, with annotated with ontological terms, and systems can know what each term means. I will go more in depth about that in a minute as well. So the main elements in this RDF are the adverse outcome pathways, key events relationships, based on the main pages that are on the AOP-Wiki as well, but also the stressors, chemicals that are in them, taxonomy, organ and cell terms. After we converted this AOP-Wiki website into RDF, we put it into a SPARQL endpoint. Docker image. So this is a tool that we deployed in the OpenRiskNet E infrastructure. It is a Virtuoso Docker image loaded with AOP-Wiki RDF data, which is available from the link there. And it allows querying the data in many ways and finding connections that you cannot do in AOP-Wiki as it is right now on the website. I have put some example queries, for which I will ask a bit of assistance from Thomas. In the description underneath this slide, there's a link to the service. So this is the general SPARQL endpoint page, and this is the place where you write the query and extract the information from the RDF. Also in the description underneath the slide, I've written 4 queries, of which we will probably do only 3, that we can just try out. If you could try to copy one of them, at least the first one. Yeah, it's a bit hard to see on this. I was hoping it worked on my laptop directly. Okay, now you copied, I think, the first 2 queries. Yes. So for example, this query that you have in your screen now, if you run this, it gives you all the measurement methods for a certain AOP. In this case, it's AOP number 12. It's just a random number that we chose to show what this tool what this RDF can do. So this kind of querying is not possible on the AOP-Wiki as it is right now, but making it RDF in a shape like this allows querying the data in many different ways. Can you try the second query as well, Thomas? The second query is written to provide all key events that could result from a certain chemical. In this case, the chemical that we chose was allyl alcohol, which is one of the stressor chemicals of the AOP that you saw before, which was the AOP by Brigitte Landersman, protein alkylation leading to liver fibrosis. And depending on what we ask as output, we can get all the information that we want from this. So from the chemical, we went to the adverse outcome pathway that it is involved in, and we took from this adverse outcome pathway, which can be more than one, by the way, all the key events that are involved, and which could basically be effects of this chemical. Of course, this is just a simple example, but if the database would get more elaborate and more entities, more AOPs get added, the modular effect of the adverse outcome pathway wiki could really shine with queries like this, in which you can make these connections from a single chemical to all the key events over multiple adverse outcome pathways, over multiple connections that you can make. The third query that I wrote was a query to get all the molecular initiating events that could lead to a certain adverse outcome, in which I only chose a simple example as well, being an adverse outcome on liver steatosis, for which there are, as visible in the screen, 2 molecular initiating events present in the AOP-Wiki that lead to this adverse outcome, who are both part of the same adverse outcome itself, of an adverse outcome pathway itself. So all these pages, all these clickable links that you see there are the actual links to the website of the AOP-Wiki. And the black text is just the description, the titles that these pages contain. The last query that I have on the website, on the page, is a federated query, which could take a little bit more time, but I think it is worth to try. Let's see what time we have time. It is a federated query in which we take all chemicals from the AOP-Wiki. We go to Wikidata to transfer it into a different type of identifier. We don't use BridgeDb in this one yet. And we go to WikiPathways, a molecular pathway database, which also contains a SPARQL endpoint And we ask to this database for each chemical in which pathway this chemical is located. So could you try to run this, Thomas?
It is running, but it takes a moment.
Yeah, so it's always a bit on the edge. This Virtuoso SPARQL endpoint right now has a limit of 30 seconds. And if it goes longer than this, it will just fail. But if it's shorter, it will make it. And I had a screenshot backup if it doesn't work, so I can put it in a slide. Oh, it is actually working. Great. It's always about on the edge of making it or not making it, because it takes, of course, a while. Because this SPARQL endpoint goes first to Wikidata, which is a database with many types of data, but it has to enter it from the outside and use this result that comes from that to enter another SPARQL endpoint. So there's two different connections that needs to be made for each line in this case. And this shows that this SPARQL endpoint can provide you a lot of information just from one single query from different databases. So we start at the left side with a chemical. We see in which adverse outcome pathways they are part of. We go to the CAS numbers, the CAS identifiers being the chemical identifier, and on the right side we can see the pathways that they are located in. And all these all these blue links, they are actually the connections to the pathways that they are in. I guess you could click or try to click one of them. For example, yeah, you see there booty rate. It is apart from the from one of the AOPs that you had, and it is present in this pathway on WikiPathways. Of course, we did not filter here for animal, which we also didn't do in the AOP-Wiki. So bear with me on that. That filtering on taxonomy is a step that we of course need to add. But that's something that's also not easily done yet in the AOP-Wiki as well, so we cannot take it in account for WikiPathways here. But of course, that would be a great addition to have. Okay, thank you. So the next tool that we worked on to add in the Wiki in the OpenRiskNet infrastructure is BridgeDb. I mentioned it before. It is a bridge between databases. It has mapping files that can convert any type of identifier for a large variety of species of genes, for genes to, sorry, let me rephrase. It has the capability to map identifiers for genes and proteins for a large variety of species, as well as metabolites, genetic variants, and metabolite interactions. So this tool we also Dockerized, so to say, and integrated it in the E infrastructure, which I also would like to show. So can you go back to the can you click on this link and go to the notes? In the notes, I wrote the two things that you need to add in the in this Swagger interface. So there's the data source, sorry, and there's the identifier. And in this example, I want to show that we can use this tool with the first call indeed on this page. Okay, you click it, and then on the right side, try it out. And there, no, this one you keep, sorry. The L, the one that says system code, you change that to the CA. It's a bit on the bottom. Yes, and there you put the CAS registry number that you just showed, which is the one for valproic acid. And when you execute this, this returns to you the list of all the identifiers that are from other databases a bit below, that are mapped to this valproic acid chemical. And they all make sense. All the identifiers that are seen here, they are accessible in these other databases and provide you with all the extra information that these databases are made for. And besides this user interface, this tool is also accessible through simply entering an URL in the browser, which returns you just the list that you just saw, but you can immediately use. For example, if you would enter it from a Python notebook, you can get a result and immediately use it to continue your analysis or to continue your workflow. So if you would paste this in the browser, and it would give you the result that you just saw on the user interface as well. Okay, so the next tool that we added to the repository of OpenRiskNet is the WikiPathways. WikiPathways is a biological pathway database, which contains molecular pathways, so therefore molecular knowledge. And the way it is created is that it is interoperable with many different data sources. Besides the identifier uses that I already was already displayed with the query that was done from the AOP-Wiki, it contains itself the SPARQL endpoint and uses ontologies to describe what is inside these pathways. Also, WikiPathways is used as a base pathway data set in PathVisio to perform pathway analysis, and can be used in Cytoscape for network analysis. To go more into the analysis of omics data sets. I will skip in this one the demos, but I can just describe what it is. It has the same user interface as the AOP-Wiki has, because it has the same base image, the Virtuoso open source 7 Docker image, and then this one is loaded with WikiPathways RDF. And for this one, all the deployment files are present and available through the link there, which you can enter if you have the slides later on. It's not necessary for now to go into that. If you're interested in how to set up this SPARQL endpoint and load it with the data. And this database also allows you to do many types of queries to look for example, all genes genes in a particular pathway, or give you all pathways for a particular gene. The same way you can create your query in any way you want. The right side of my overview that I had in the beginning was on the left side AOPWiki, on the middle BridgeDb, and on the right side the databases. And one of the main tools in OpenRiskNet that provides access to databases is the Edelweiss Data Explorer, which allows querying for data sets and assay and results for databases such as ToxCast, ToxRefDB, and gggates. On the below, you see the user interface of this tool, which is provided by Edelweiss Connect. I will not go into this in any more detail, but this is this tool also has APIs to make direct calls from, for example, a Python script to enter your identifier that you have, your gene, your chemical, correct me, and ask all the data from these databases that result from exposure to this chemical. The status of AOPLink at this moment, of course, this is just a progression webinar, is that we are close to answering these research questions. So the research questions were, can we find data that supports this adverse outcome pathway? And the other question was, which AOP can be supported by this certain data set? So the two directional question. And for that, we needed these tools, these couple of services that we needed to answer these questions. And they are all deployed. They are all working in the OpenRiskNet infrastructure. However, these tools separately do not do anything yet. You can manually go through the few of them and you can make your answers from there. But in OpenRiskNet, we want to make these tools parts of workflows. So that's the next thing that we are going to work on. So that's my this is my third section. What are the plans for 2019? Refinement and coverage. So the AOP-Wiki, as I mentioned before, we converted in RDF, but still some elements are missing because we can either not annotate them, or it is very confusing to model this in a triple format. So things to add still in the next few weeks or months from now are, for example, the key event components being ontological annotations of key events, as well as the biological levels. Because as I told before, all key events of all AOPs start with key events on molecular level and go higher in level to the adverse outcome on the organ or individual level. Besides, there's a couple of additions that we could add to this AOP-Wiki. For example, we can use identifier mapping for HGNC genes that are described in key events. So we can identify the genes that are described. As well, use information from, for example, the stressor AOP, being SAOP, which has an annotated list of key events related to toxicast assays, which they manually created themselves. Besides chemical target interactions at the molecular initiating event. Also here, BridgeDb functionality can be included, which would allow more types of identifiers, for example, for all these stressors, for all the chemicals that are inside this database, that help in making this data source more interoperable with other places and replacing a mapping tool in between. The other part that was kind of an extension of our basic overview was the pathway analysis. For that, we wanted to use and integrate PathVisioRPC. PathVisio is the pathway analysis tool that uses WikiPathways as a base pathway dataset and allows the analysis of omics datasets. And it's this tool that is described here is PathVisioRPC, which contains the functionalities of PathVisio to analyze data, but without needing to install the software. And it's accessible from many types of programming languages. And it can be used then to not just analyze the transcriptomics data or any other type of omics data, but also visualize it on pathways and make figures from it with expression levels on it, on the different individual nodes. So this is a plan that we want to add in this scheme as well, to make not just the research question of linking data to an adverse outcome pathway, but also continuing and seeing if we can actually analyze this data directly. The last part, that's actually a really central tool in this workflow as well, is one of the winners of the OpenRiskNet implementation challenge of the end of last year, which is the AOP database. As already mentioned in the beginning, Holly Martens from the US EPA and others are responsible for the development of this adverse outcome pathway database, and they have a database of adverse outcome pathways related to the AOP-Wiki that contain many types of information connected to adverse outcome pathways, such as the genes, proteins, pathways, toxicast data, and diseases that are related to them. So this tool is something that we would also like to integrate into our workflow system, because it's they contain large amount of data that can be useful for these workflows as well. And next week on April 8th at 4 p.m. Central European summertime, the Holly Mortensen and others from the US EPA will actually present in a webinar similar to this, the AOP database. On the bottom right, if you have the slides, you can click the two main references that are covering the AOP database. So here we are back to the original overview that I showed before. And with the next few slides, I show you how we can even extend this more. So can you go one more slide, please? For example, I mentioned the AOP database, which is containing a lot of information about AOPs. We need to figure out how it fits with more connections, but it is already, we can see that it can be an essential part in this case study, as well as the SAOP, which I mentioned before, which connects key events to toxicast data. And if we want to go even more in detail and extend with more tools that are available in OpenRiskNet, we can, for example, look for chemical similarities for stressors that cause the molecular initiating event using the ChemID converter and Lazar, which are both tools integrated by other partners in this project, and which are directly usable, for example, through Python notebooks. Also, Skyview is a tool that is used for text mining, reading text, which could give us a great amount of information from the description that are actually present on the AOP-Wiki. Like I said, a lot of content on the AOP-Wiki is free text descriptions. Of course, from there we can extract genes, proteins, pathways, many types of things, diseases. But this tool does that, is able to do that automatically and provide the information from the text quicker than the human can read. On the other side, there's also extra databases that we would like to see added to this workflow. For example, the Area Express, the Geo, and the DIXA omics databases that contain a great amount of data sets, which can be an important player in the eventual pathway analysis that we would like to include with PathVisioRPC. This is my last slide. Of course, all the tools separately have already great functionalities, but if we would combine them, we can make these nice workflows and answer complex biological questions that could support the risk assessment community. So for this case study, we will use Jupyter notebooks. And OpenRiskNet has a Jupyter Hub service deployed on OpenRiskNet infrastructure, which allows spawning of many types of preloaded Jupyter hubs, and are useful to enter notebooks and answer the questions that are there. I wanted to put here also an example of a notebook, but I didn't, so I will still put it in the slide afterwards. I would like to thank you all for listening in tonight, and sorry for all the technical issues here. If there are any questions, I would like to answer them to you. Thank you very much.
Okay, thank you, Marvin. That was really great, I think. I hope that everyone agrees. If you have questions, please raise your hand virtually, and then we can open this floor to you, or you can also type it in the question section if you prefer that.
Hi, do you hear me?
Yes, sorry, I had a little bit of technical problems. We can hear
it was my microphone that was muted. Vincent from Maastricht University, also the Institute of Data Science. I wonder, do you plan on publishing the SPARQL queries on GitHub. Like you showed a lot of different SPARQL query on the production Virtuoso, and I wonder if it's possible to get them on GitHub, because it's easier to get them and play with them.
Yeah, sure. I can put them on GitHub right away, and I sent the link to you.
Yeah, in general, this is the concept that we will make these workflows, the Jupyter workflows and the queries available. Everything will be described, and this is exactly what we try to do, is giving the people the chance to learn from what we designed and then extend that into the directions to answer the real questions. But everything which we present, the slides will be available, and we will create more and more workflows, Jupyter workflows, as well as queries, or most of the time these queries are then part of the Jupyter notebook. Therefore, this will all be documented and will be available on the website.
Perfect. Thanks a lot.
You're welcome. Good. Then I think, I don't see any more questions, no raised hands anymore. Therefore, thank you everyone again for joining, and I look forward to welcome many of you again in 2 weeks' time, or would like to see your feedback on the questionnaire or on any other email, or use our support system. If you have any questions, please do not hesitate to contact us. Marvin, thank you very much again for your presentation, and talk to you all soon. Thank you.
Thank you. Bye-bye.